All right, so mechanisms of membrane transport. There's not a diagram for that. So yes, we have diffusion. Yes, there's osmosis, right? There's facilitated diffusion, right? Then there's different kinds of transport, active and passive. All of these terms, do you know the difference, right? And it's not just, for example, this is one I really like because it's so important to the cell. Glucose is water soluble. You know that because when you stir a sugar into water, you can't see it, it dissolves, right? So because it's water soluble, that means it can't go through the membrane. But your cell needs glucose to make ATP, a lot of it, right? So naturally, the cell would want to have a membrane transport mechanism that would help a lot of glucose come into the cell, right? But it doesn't make sense to bring glucose in to make ATP and use ATP to do that, right? So what is the mechanism that brings glucose into the cell?
Insulin.
Insulin is a chemical, but what's of all your...
Active transport.
It's actually facilitated. It's facilitated transport. That's where, well, you can check it out. The glucose molecule just has to dock with a little receptor, and the receptor opens and brings it in. No ATP required. Okay. So you'll have these questions about specific transport mechanisms, and then more general ones. Passive transport, no ATP needed. Active transport, you need ATP to move that material. And the active transport, an example would be your conduction of your electrical signals on the nerve membranes. Those are electrons moving down the surface, right? And when you do that, the electrons get trapped on the wrong side of the membrane after your action potential moves down the cell. So you actually have to pump back the ions that help you send that nerve signal back to the right side. You go in and look at that, right? You got to be a little bit more than term and definition. You should have an example that would apply to it so you can really understand how that's working. All right, intracellular attachments. That would be from this diagram right here. And there are basically these three kinds of attachments. Occluding means, well, it's the opposite of including. I don't know why they don't call it excluding. Right? There's no materials between the walls right there. They're like staples. A gap junction has these pores in the connectors. The pores are great for what?
Because we might need that space when this goes up.
Yeah, for moving things through, for transporting things through from cell to cell, okay? And I gave you an example about how the cells of the heart, they start a contraction of the atria in one part of the heart, but both atria contract at the same time. That's because the electrical signal through ions have moved through these channels through all the cells of the atria. That's one example. And then finally, these anchoring style connectors are like Molly bolts. They not only connect the cell walls, but they brace themselves with these plates on either side, and that makes these very strong for attaching the cytoskeleton. This would be along the wall of a cell. Here's down at the floor of the cell, the basal level. So I've translated that diagram into, are you ready? You guys are like the paparazzi. I kind of feel like Angelina Jolie, except for every camera's away from me. Here you go, that's what it looks like on a test. And again, don't just go, okay, B is these gap junctions. I may not even mention gap. I might say these junctions are particularly helpful in the heart for transmitting, the conduction for a heartbeat, right? So you got to be careful about studying, right? Get a more complete idea of how this all works. All right, so much for intracellular attachments. What's next on our list? The cell life cycle, which is including your interphase and your various phases of mitosis there. I think I mentioned, nope, we gotta mention that now. Is that you, Chris? Don't forget you have to take a makeup test. Did you miss one when you were being added? Okay. All right, so next, no. Where'd they go? Yep, there they are. Okay, so this is the diagram that's in your book. And your diagram nicely provides both an illustrated version and the histology version. I'm gathering that probably all of you, I didn't get one question about the whitefish blastula yesterday and Tuesday, right? Most of you in lab didn't look at those slides, okay? So the diagrams will be on the lecture test, but in lab you'll actually see mitotic figures, okay? So you're going to have to go back, and if you didn't see them in real life, you can go to 170. They have a microscopes and slides in there. They'll be happy to help you with that. Or you can check these out online and hope you can transfer these images to the ones that I use. Okay. I mentioned the other day in lab that I don't think my images are staying purple. So if that's going to throw you off, then you're going to have to get a more generalized idea. Okay. And you can always practice. Go online and Google mitosis histology, and you can see everyone's stained versions of these, and just keep practicing until you recognize them, no matter what stain or style they're presented. So that's so much for the recognizing each phase of mitosis. There's some other key points. For example, after which phase does cytokinesis occur?
Telophase.
Telophase. And what is cytokinesis?
Cells.
Cell moving, right? Kinesis is like kinesiology, moving, the cell moving away. Where is the DNA and the organelles and all of that replicated?
Interphase.
Interphase. That's a very common misconception by students. DNA is already replicated when mitosis starts. Okay? There's already twice as many chromosomes. All right, and then the rest, of course you'll get these figures in a small statement, right? Okay, just like I normally ask you questions. Well, you can see there's a lot of exciting things on this test. I can hardly wait till you take it. I'm not showing you microscopy, okay? But I've talked to you all along about the 3 different styles that we're used to, right? The light microscopy and the 2 electron styles. Should be able to recognize an image, whether it's light transmission or scanning electron microscopy, and how they're all done, okay? How they're all set up. I'm not going to show you that because you'll take pictures and memorize it, and it's really not a fair test. That part of the test is also me gathering data for you, and you're learning about your learning objective about microscopy. If you look on your syllabus, I have these 6 learning objectives, and I've got to teach you and test you on those. Okay, so cellular respiration, which is ATP production. If you don't learn that equation, you're going to continue to lose points throughout the entire semester. I'll never give up on asking that. So if all of you just memorize the equation, then you always get all the points every time I ask it. The thing of it is in the past people have failed to do that. So I keep asking. What do we start out when we make ATP? What are our beginning resources?
Glucose.
Glucose.
Oxygen.
Oxygen, right? Glucose plus oxygen. Eventually, you break down the glucose. You separate out every carbon from the glucose. It's a 6-carbon sugar. And as you're doing that, you're kicking out ATP a little at a time. So what does that eventually end up with? Carbon dioxide and
energy.
Water, right? That's the chemical part of it. Now the energy part of it. What do you start out with? ADP plus phosphate, and you make ATP. I definitely type that right into your notes, okay? Learn it because I'll ask you several times. That's a huge concept, and you're going to get a lot more lessons on it when you get to physiology, like how it actually works. Okay, now moving on to the tissues here. These are the 4 types of tissues, their characteristics and functions. Okay, so there's several questions about, well, first there's a few questions just sorting out the 4. Okay, epithelial, connective, muscle, and neural, right? And your epithelial and connective tissue, thank you there for the backup, someone, because help me out there. Those 2 are the most complicated, right? There's several kinds of epithelial tissue and connective tissue. So you need to look into their characteristics and their overall functions. For example, which one of the tissue types is great for coverings and linings? Epithelial. Which one is great for electrical communication throughout the body?
Nervous.
Neural, right? Which ones are great for contractile mechanisms? Muscle. Muscles are contractile cells, right? Okay, then you get into each one of those types. And in the connective tissues, you have connective tissue proper, which is packing the body, but then you have fluid and supportive, right? What would be an example of fluid connective tissue? Blood or lymph, and supportive? Bone and cartilage, right? Okay. Those are all easy. The connective tissue proper is a little bit more complicated. If there's dents in the name, what is that describing? What kind of fiber would that be mostly made of? Collagen. If it's irregular, how is that collagen arranged? Net-like or disorganized. So you have irregular connective tissue in your reticular layer of your dermis, right? Thick collagen fibers in all directions, okay? So, okay, what does that regular configuration mean about how the forces are affecting that tissue? Parallel means one direction, that would be regular. Irregular is pulls in many directions, okay? It's the same application for just a collagen fiber. Those are usually, if they're parallel, they're used to pull at each end, right? If they're irregular, then they pull in multiple directions. Right? Reticular pulls in multiple directions, it's net-like. Okay, so do you get the idea about your knowledge? It's not going to be your standard test. I'm looking for you to understand concepts. And I've been trying to present general themes that you can use. There's 100 questions. You get the whole time. Everyone finishes in the past with plenty of time. I have people leaving in half an hour. And then people staying the whole hour and 15 minutes. All right, so then you get into cells and fibers. Okay, so we did that little thing about how you name cells, fibrocytes, osteoblasts. Do you know those parts of a word that help you understand what those cells do? I think there were also Mast and Merkel. There's a whole list that we went over in your lecture notes, that chapter. And of course, I'm going to be going over the fibers. Those are collagen, reticular, and elastic. But am I going to say, what's the name of the fiber that it can recoil and it's shaped like a telephone cord? What would that be? Elastic. Does it sound like that's the kind of question I would ask? No, I would ask, what would be a good fiber to use in your ear? Elastic, right? Okay, I'm just kind of warning you here. You have a couple weeks now, so you got the whole concept about the kinds of questions I ask. Now, mechanisms of secretion. I think we have a diagram. Oh, this will work. Okay. And did I actually include? No, I didn't actually include the figure out of the test. Now, these are 3 different ways that cells create secretions: merocrine, apocrine, holocrine. And I like those different hints that I gave you, that Apocrine, the apical surface comes off. Holocrin,